A very good morning, students. Now we come to a new chapter in the genetics, and in series of these lectures, now we will deal with the modes of inheritance and the gene disorder. Okay, this is an important topic. Now, since uh, last lectures we were on the chromosomes, and I told you the various aspects of the chromosome and the abnormalities which were associated with the number of the chromosome, that is the numerical abnormalities or that of the structural abnormalities of the chromosomes. Okay, but from today we will be learning about the genes. Okay, the I mean to say, what are the abnormalities which are associated with the gene disorder? That means a gene mutation we will. And if the gene is abnormal or defective, what disease it will cause? Okay. Now directly we are jumping from the chromosomes, which were a microscopic structure, okay, which we can be seen with the help of the microscope. Now we are coming to the genes which are not the molecular structures, okay, which are sitting of course on the chromosomes, but we cannot visualize these genes. Okay, why I am doing so? Because most of the portion of the genetics which deals with the molecular structures of the gene and various I mean to say associations of the gene, okay, they are in the biochemistry, not in the anatomy. So I have just left that portion, but this portion is in your curriculum. Okay, let us see what is gene. Okay, though it is in biochemistry, I will have to define you what is a gene. A gene is a I mean to say I am defining a structural gene which are more common, which are responsible for the formation of the protein. So a structural gene is defined as a segment of DNA. That means you know the double helix of DNA, which forms the chromatin material I have told you during the chromosomes. Okay, so it is a segment of the DNA which contains the information, or information means what information are stored in the DNA strain. It is the code, or it is the code for synthesis uh, of one complete polypeptide chain is one structural gene. And as I told you, there are about 30,000 genes which are present in human genome. Okay, total 30,000, still less than that. Okay, then so a gene is that part of the DNA molecule which is having the code, or which can which is having the information, okay, or which will guide the formation of one complete polypeptide chain. Okay, that is called as the structural gene. So I'm not going into the genes which will be taught to you by the biochemistry peoples. Okay, I'm also not going to tell you about, because it is not in anatomy codes, that is the genetic code. I am not going to tell you how genetic code are formed, what is transcription, what is transcription factors are there, okay, how the genes are regulated, these structural genes, how they are regulated. I will also not tell you about the replication. So we are directly here on the gene disorder, and to learn the gene disorders, it is necessary to learn the modes of inheritance, how these genes are transmitted from one generation to the next, which are sitting on the chromosomes. Okay, so they follow some modes of the transmission, okay, which are called as the modes of inheritance. So with this introduction, I am going to the next slide, which will tell you about the modes of inheritance. Now, modes of inheritance, okay, to understand the gene disorder, our main aim is to understand the gene disorder. What are those genetic diseases due to abnormal gene or a mutant gene? Okay, these can only be understood, okay, it is important to know the pattern of inheritance first. Okay, so just to understand any genetic disease which is determined by the abnormal gene, it is most important to know what is its mode of inheritance. Okay, whether that disease is a dominant disease, recessive disease, or sex linked disease. Okay. Hmm? So this thing has to be known. What do you mean by dominant? A dominant is disease is expressed, or otherwise also, let us see, not disease, but a normal dominant character is expressed when the gene is even in a single dose. Single dose means otherwise the gene, they form the pair, allelic pair, which are located on one particular location. See, for example, in human being there are 46 chromosomes which are present in pairs. Okay, so there are how many pairs? 23 pairs. Out one member of this pair comes from mother and another member of the pairs comes from father, but they contain exactly the same gene which determine a particular trait, and they are located on the same location, on the chromosome coming from father and chromosome coming from mother. Out of these two, so that means for each trait or each character, whether normal or abnormal, there are two genes, means a pair of gene. One is sitting on paternal chromosome, another is sitting on maternal chromosome. Okay, even if one chromosome when is responsible to express the trait, okay, in a single dose, then it is called as dominant. Okay, it is not that both the genes which are located on at a locus on a homologous pair of chromosome both should be active. Even if a single gene is active and if capable to express that trait or character, it is defined as dominant. Okay, the recessive the recessive character, let us see here. Both the genes are required, which is determining a trait, as we have learned in the Mendel's experiment, that the trait is the height of the plants. Okay, it may be short or the tall. Okay, so if for every trait there are the pair of genes is there, and this pair of genes are responsible for that trait. So in the recessive case, both the genes are should be active. Then only the trait is expressed or it is shown physically. I mean, say phenotypically, it is shown. So here both the gene have to be active when they are those which are present on homologous pair of chromosome. Right? This dominant and recessive is true for the autosomes, which are 44 chromosome, 22 pair, and this is also true in case of the sex chromosomes. Okay, they are called as sex linked. Okay, now these two X chromosomes are there on the female, and there it is again the same, dominant and recessive. It may not be that effective in case of male because chromosome are not homologous. They are exactly not of shape or size. Okay, so this is the dominant and the recessive explanation. Now, why it is necessary to know the modes of inheritance? It is necessary to know the modes of inheritance for any normal trait or that of any abnormal gene expression for accurate diagnosis of the genetic disease. Number one, it is for accurate diagnosis of genetic disease. Second, for the calculation of the risk of occurrence. Calculation whether this trait will express in the second child, third child or not, and to suggest the way how to prevent it. That is the importance of knowing the modes of inheritance. Okay, now I pass to the next slide. Now the inheritance follow the following pattern. What we mean by inheritance, I have already explained, just repeat it. Inheritance means when the characters are transmitted from one generation to the next generation. Okay, that is called as inheritance, and this transmission of character follows the following two patterns: monogenic and polygenic. Monogenic means single gene inheritance. Okay, it is the single gene. When character is transmitted by a single gene or determined by a single gene, that is called as the monogenic inheritance. And the Mendel's experiment, or Mendel's, what he did was only based on the characters which were determined by the single gene. Okay, so this is also known as Mendelian inheritance. Okay, so the monogenic means the characters which are determined by a single gene, and this is also known sometimes as Mendelian inheritance. That way, the Mendel was lucky to select only those characters in pea plants which were determined by single gene, because he was not knowing at that time what is polygenic inheritance. Okay, because nothing was known about the gene. Okay, let's come to the second type of the inheritance, which is called as the polygenic inheritance, or the multiple genes when are involved, not by a single gene if a character is determined. Many characters in our body are in human beings, they are determined by many genes. Okay, not by a single gene but many genes, and they follow the different patterns of inheritance. This we will not go further in polygenic inheritance. What we are going to learn in the next few lectures will be based only on monogenic inheritance or single gene inheritance. Then there is another inheritance, what is called as multifactorial inheritance. And multifactorial means not only which is determined by the gene, multiple genes, but also the environmental effect which plays a role in its expression. For example, okay, diet. Okay, if one is taking a good nourishing diet, then his height will be more, beside the genes which he got from his parents. Okay, then there are the various environmental factors and chemicals. They also ultimately affect the expression of the gene. Okay, now thus we came to know that there are the two types of the inheritance: the monogenic and the polygenic. Going to the next slide. Now this monogenic or Mendelian inheritance, so we will now confine only to the monogenic or Mendelian inheritance. In this kind of inheritance, characters are determined by single gene, that I have already told you. This kind of inheritance follows the Mendel's laws of inheritance. Okay, so we have already learned about the Mendel's laws of inheritance. Okay, now the single gene inheritance or Mendelian inheritance is of two types. They are of two types. And autosomal inheritance means inheritance due to the genes which are present onto the autosome, those 22 pairs of chromosome, 44 chromosomes. And then second category in the Mendelian inheritance or the single gene inheritance is sex linked inheritance. Sex linked inheritance, that means those genes which are present onto the sex chromosome, that is the X and Y. Okay, X and Y, these expression of these genes which are present on sex chromosome, they are called as sex linked inheritance, while the rest of the other 44 chromosome are 22, since they are autosomes, it is called as autosomal inheritance. Okay, now the autosomal inheritance is further classified as autosomal dominant and autosomal recessive. That means gene which are present on autosomes, okay, those 22 pairs, they may express either in single dose, then it is called as autosomal dominant, or in the double dose, that means both the genes are necessary to express, hence it is called as autosomal recessive. See this. to just determine the what kind of inheritance a person is suffering from an genetic disease, or what is the inheritance of a normal gene, okay, whether it is autosomal dominant or autosomal in I mean say recessive, or sex linked dominant or sex linked recessive. One has to take the history of that disease or that character in the family, okay. So whenever you will get a genetic person or a patient with the genetic disease, then you have to ask the history of that disease in that family, okay. And you have to make a figure, okay, or a diagram of the history of that disease, which is necessary just to determine whether this inheritance was autosomal or recessive, okay, autosomal or recessive. And this diagram, which is just plotted by asking the history of the family, is called as pedigree chart. One of the pedigree chart and its symbols which are used for formation of pedigree chart, I will very quickly just tell you, okay. So what is pedigree chart? It is a diagram which shows the ancestral history of a group of relatives, okay, relatives, okay. You will have to ask a detailed history. And in this pedigree chart, one pedigree chart is shown at the bottom of this figure, and this is showing only two generations, that is parents and their offsprings are shown here. See these children's, and here these pedigree chart use some symbols, and let us see. A male is always represented by a square, and if he is healthy, not suffering from that genetic disease to which you are investigating, then it is an empty square. Healthy male is empty, while a male which is affected from that disease to which you are investigating, then it is a solid. You may use any color. I have used this color. You may use any color, even the black, okay. If you are drawing it with pencil, fill that square with the black pencil, okay, color. Then the female is a circle. A healthy female is always represented as circle, and affected female by a solid circle. A mating, okay, it may be a marriage or a sexual mating, okay, which has resulted into the offsprings or the children, is represented by a line joining a male and a female, okay. So this shows the mating, okay. And then if the mating is between two cousins, first degree or second degree cousins, then you have to represent them by the double line, which has a special importance, because the current I mean to say the this cousin marriage is very important in the genetics to know. If you will not know the history about the cousin marriage, you are going to miss many things, okay. Now this solid square with an arrow mark indicate that this is the first male in the family to be identified from that disease, okay. First male to be identified from that disease. And then a solid circle will identify a female who has come to be under the notice of investigator as the first person to suffer from that disease. For male it is called as propositus, and for female it will be called as proposita, okay, proposita. Now when a square is crossed oblique by an oblique line, it indicates the death, okay. You have to represent the death of the I mean say relatives, okay. Don't miss that. If oh he's no more, so why should we draw in pedigree chart? No, that is more important, okay, to represent the death. Uh, Now this half of the square and half of the circle, it indicate the carrier state. That means the person is having that a mutant gene, abnormal gene, though it may not be expressed as a disease. If it would have expressed, you would have drawn this square or this circle as solid, but it is half solid, indicating that that female or male is a carrier. And this is the first, I mean to say, I mean, first generation is shown here, a male and female which have the marriage or mating, and then they have the three children. uh, First, from left to right, you have to draw this in sequence, whether they are male or female, and by Roman letter, that is first generation and second generation you can, and third and fourth generation you can keep on writing, okay, on one side of the pedigree chart, okay, pedigree chart. And thus a pedigree chart is prepared, and we will see just that it is very important to draw, because this will give you, just by looking at the pedigree chart, the mode of inheritance, whether this pattern follows the dominant or the recessive inheritance. Okay, passing to the next slide. Now we come to the autosomal dominant inheritance. With this background, now you are at the autosomal dominant inheritance. What does it mean? It means that the gene is sitting on autosome. Those 22 pairs are 44 chromosomes. Okay, and the gene, even in a single dose, will be expressed. Okay, it will show the disease, or the normal character is determined by, even if a single gene is there, that is called as the dominant gene. Okay, autosomal dominant disorders occurs due to presence of single mutant gene. Mutant means abnormal. I will take a separate lecture on mutation, and this is a single mutant gene, means single abnormal gene on one autosomal chromosome of the pair. As I have already explained, only out of the pair, if one gene is active, that will be expressing, and that is autosomal dominant. Now following are some common autosomal dominant disorder. Various disease, genetic disease, they are autosomal dominant in nature. Okay, Huntington's disease, that is Huntington's chorea, also called as neurofibromatosis. I'm going to tell you about this disease, though it is not in your course. Okay, there is only one disease from the autosomal dominant list which is in the course, is the achondroplasia. Then the polycystic kidney, the osteogenesis imperfecta, okay, brachydactyly and polydactyly. I will not go into the details, and we will learn about the two diseases from autosomal dominant inheritance, achondroplasia and neurofibromatosis. Going to the next slide, and this is very important slide. You note down, if those who are not having the books, you note down in the your, I mean to say, just for your analysis or remembrance of the characters of autosomal dominant inheritance. What character will be followed if a gene is autosomal dominant and it is expressed as a disease or a normal character? Okay, these will be there. Okay, so many points are there in the characteristic. Males and females are equally affected on the autosomes. Okay, because if in a pedigree chart you see that, oh, affected are males also and affected are females also, then it means that this gene is sitting on the autosomes. Otherwise it should be on the sex chromosome, either X or Y, but it is both males and females are equally affected. That means it is, the gene is autosomal. That is the biggest finding which you will have to know, whether the pedigree chart which I am seeing, it is indicating an autosomal or the sex linked inheritance. So if both are equally affected, that means it is the autosomal, right? Gene is on the autosomes. Disorder seen in every generation without skipping, because it is dominant, and if it is dominant, some of the children in that pedigree chart in every generation will be suffering. Okay, and there will be no skipping. Okay, no skipping, right? An affected person will always have an affected parent. That means all of a sudden a person will not be affected whose parents are normal. He will always have an affected parent, either mother or the father. Okay, and will be affected, and then only the disease will go to the children. Now, if in case all of a sudden you get an affected person, okay, and subsequently in the subsequent generations there are the affected people, then what it indicate? It indicate that he is the first person in the family to have this disease, and he will be called as if male proposita, okay, or female propositus, okay, the first person. And this also indicate that the mutation or abnormality in the gene must have occurred in the parents of that person, okay, who is the first to be shown, first to be showing those this disease, right? Now the normal person will not transmit the disorder to the offspring. Okay, if suppose in a family two brothers are affected and one sister is affected and the rest of the other brother and sisters they are unaffected, then those who are unaffected, not showing the disease, they will never transmit to their children. Okay, because they don't have that abnormal gene. They don't have that abnormal gene in their on their chromosomes. Okay, so you can tell to your I mean to say patient that if you are having two children, one is affected, the affected children will never have this transmit that I mean say this disease will never come to his family or her family, because she or he is normal. Okay, normal. That is very important to tell to your patient. Okay, number of affected and normal persons are equal in each generation. Now if a large family or pedigree chart is your large showing many generation, and if you will calculate how many are affected and how many are normal, it will always come close to 50%. Okay, 50% affected and 50% normal. This is important in the sense that because this is determined by a single gene on one of the chromosome of the pair. That means 50% gametes will be normal and 50, because one chromosome will go to the 50% gametes and other chromosome which is affected with the affected gene will go to the remaining 50% of the gametes during gametogenesis. There is no carrier state in the inheritance as we see in the recessive case. Okay, when we will come to the recessive, I will tell you this carrier state. There is nothing like carrier. Carrier means when the recessive character is there, they are not suffering. It means only one gene is abnormal gene but not expressing. Okay, that I will tell you in detail when I will come to the recessive. Right, these were very important characteristic of the autosomal dominant inheritance. Right. Now this is the pedigree chart of an autosomal dominant inheritance. Right. This pedigree chart shows that father is affected because it is solid here, and then he has one daughter, second daughter, third is the male child, and fourth is male child. Out of the four child which is having, okay, two daughters and three sons, no sorry, one daughter and three sons, one daughter is affected and one son, means 50% of his children are affected. And now the one daughter and other son is unaffected. Okay, yes, there are two daughters. Okay, sorry, there were two daughters here, one affected, one normal. There were two sons, one affected, one normal. Okay, so the 50% out of 450%, and it is being transmitted equally to the, I mean say, to the both the sexes. The daughter is also affected as well as the son is also affected. Okay, so this is a indicating that it must be a gene sitting on the autosomes. Okay, nothing to do with the sex of a person. And then this daughter marries to some person or male from outside, and then she will have the, out of three child, two are suffering, one male and one female, and this male who was suffering will have four had four sons, two sons and two daughters, and again two are sons are suffering and daughters are normal. Okay, but those this daughter and this son of this parents, they were not suffering, so they will not transmit this disease to their family further, and never in future in the grandchildren's, okay, or the great grandchildren will not be having this disease, because that gene is not being transmitted from his great grandfather, okay, to this family, right? So this is just by looking at the pedigree, you can see whether this is uh, autosomal dominant or autosomal recessive. I have just told you about autosomal. So you can very well just judge, because it is both the sexes are affected. There are about 50% children in each pedigree are affected. One which is not affected will not transmit to his subsequent families, okay? Subsequent. So these are the characteristic of an autosomal dominant inheritance. Let me pass to the next slide. Now, if I further explain this, okay, this is with the help of what is called as punet square. You try to learn this formation of the punet square, and when you are suggesting to your patient in the genetic counseling, okay, of course, something you will have to tell to your, even if you are not a genetist, you are a doctor, you will have to give the answer whether this disease will transmit to his offsprings, children's, and what are the chances of them to suffer. So we use this punnett square, and here we put one parent on one side, that means on horizontal side, and another parent on vertical side, okay, and then possibility, because a parent, I mean say male and female, okay, individual contain two chromosome, okay, and thus the gametes are two different types, may be possible two different types. This represent the gametes on one side, okay. So this is a normal parent, and this AA, small a, means normal genes are there on both the chromosomes. So all his gametes which will form, if it is male, the all sperm will be normal, okay. And if suppose he marries to an affected female, which is I mean say dominant, so only one gene is affected here, capital letter A, showing the one gene is affected, okay. And this is the normal gene on the other chromosome, okay. But it is expressed because it is dominant. And if you consider the matching with this kind of the gametes, or the sex cells, sperm and ovum, then possibilities are here, okay. Here is an abnormal gene, here is also an abnormal gene. So these are the, since it is dominant, okay, all these children will be affected, okay. So 50% chances are there that it is heterozygous affected patients, heterozygously affected patient. So they are they will suffer. On this side, if you see that this is a normal gene, and these were also on the normal gene, so they are, the offsprings will be born here, they will contain the normal gene which are homozygously normal, thus they are also 50%, okay. So there are chances that you tell to your patient 50% chance are there, if one of the parent is suffering, that his or her children will be 50% children will be affected, and 50% chances are that they will born normally, okay, they will born normally. Now let me go to the next, okay, and probably this is the last slide in the autosomal dominant explanation, where both the parents are affected, okay. That means one of the gene of homologous pair is affected here. A is affected gene, okay, or mutant gene is, or small a is a normal gene. And if we see the possible matchings which can be there, there will be AA, means severely affected. Both the genes are defective. And both gene will express, even when a single can express, both will definitely express, and they will express in full form, and they severely affected. So 25% are severely affected. And if you see here, one is a mutant affected gene is there, so these 50% children will also be affected. Okay, so how many will be affected when both the parents are affected? 75% will be affected. Only this 25 which contain normal genes, only 25% chances are that that the child will be a normal child. Otherwise, it will be an affected child. I think I stop here, okay? And in the next slide, I will tell you, or shall I tell you that there are two more slides, three more slides, and I will just pass it, okay? So that this will complete the whole, I mean to say, autosomal dominant. Quickly, we come to the first condition, what is called as the achondroplasia. That means the person is of the short height, and incidence of this disease is 1 in 10,000. You will see many small, okay, bone, okay, this small child, I mean say, height people you have, dwarf people you see that. Clinical features are the person is a small, of short height, head and face are of normal size, but limbs are short. They are of normal intelligence. They are not mentally retarded. Okay, just remember that. And they live normal life. Okay, and also reproduce normally. That means their functions are normal, except their height is short. Otherwise, everything is normal. Okay. What is the genetics? Okay, it is an autosomal dominant disorder. That means a single gene is a defective gene or mutant gene, and because of that this disease is there. And where is this gene located? It is due to the mutation of fibroblast growth factor receptor gene. Fibroblast growth factor receptor gene, and that means this is the gene in fibroblast which respond to the growth factor receptors of the growth factor. Okay, and when this receptor become defective, even the growth factors are available, the person will not respond to that growth hormones. Okay, and that's why it leads to the defective cartilage formation. Okay, so the formation of cartilage becomes defective, and a person is of short height because most of the long bones of the limbs they develop in cartilage. Okay, and when the cartilage itself is defective, the bone formation will not be normal, and that's the reason why their person will be of short height. But his head and neck region is normal because many of the head and neck bones they ossify from membrane, not by cartilage. Okay, or not by cartilage. Okay, now where is this gene located? This gene is located on chromosome number 4, autosome. See this, this is autosome, chromosome number 4, and on the short arm, P means short arm of the chromosome number 4 at 16.3 location. When I was a teacher, okay, long back, I mean say a teacher, still I am teacher, but when I was, okay, a few years back, okay, when I was teaching this achondroplasia, we were not knowing the location of this gene. Okay, now you are lucky to know where this gene, which is an abnormal gene responsible for the normal growth of the cartilage, is located. Okay, let's go. This is a photograph of a Jew family who migrated to Israel soon after the formation of the Israel country, and it is a big family. Okay, some of them, 7 are dwarf, and then remaining maybe 4 or 5 they were not dwarf, they were normal, again indicating that it is a autosomal dominant. Okay, indicating because some are normal and some are abnormal, and then male and female here you are seeing they are equally affected. Okay, male, so it indicates that gene is sitting on the autosomes, not on the sex chromosome. Okay, so very quickly I will pass to the next disease. And this is another disease which is not in your course. Still I am telling you, it is called as achondroplasia was in your course. Okay, so remember that neurofibromatosis incidence is 1 in 3,000 person. Okay, live birth. Clinical feature are small pea-size benign tumors. They are not cancer, but they are multiple benign tumors are present in the skin, during the late childhood. They appear during late childhood, and you must have seen many people like that. Okay, I have seen many, not as a doctor but as a general public. I have seen small pigmented skin mesopigmentation, okay, discoloration or hypercoloration, macrocephaly, large head, okay, delays, milestone. That means what are milestone? The I mean say it, the this I mean when a child is born, then he develops, and various developmental stages are there, and they are called as the milestones. Okay, and these developmental stages of a child are delayed here in case of the person suffering from neurofibromatosis, which will only appear in the late childhood. Okay, few may suffer from epilepsy, and they may also suffer from scoliosis. What is scoliosis? It is the lateral bending of the vertebral column and rotation, along with rotation. Genetics, okay, autosomal dominant inheritance with the complete penetrance, okay, complete penetrance, though I don't want to go in detail. The gene sometimes is partly expressed, sometimes it is fully expressed, okay. When it is fully expressed, it is called as complete penetrance, okay, and with a variable expressivity. That means its expression sometime is in one form and sometime it may be in other forms. Okay, so the pigmentation and the p-shaped benign tumor are its expression, which may be in different way. Okay, located on the chromosome number 17, long arm of the chromosome 17 at 11.2 position on chromosome long arm. Okay, gene codes for protein neurofibromin, which is a protein, okay, and that is responsible for the normal skin in the nervous system. I mean, say nervous tissue formation. It acts as a tumor suppressor gene, okay. And that's not in your course, the cancer genetics, but the cancer is controlled by various genes so that cancer should not appear, and there are genes and they are called as tumor suppressor gene. And when this neurofibromin is a protein which is produced due to the gene defect in the gene or mutation in gene, which is the tumor suppression gene, so that it cannot suppress formation of those benign tumor in the skin. Okay, so tumor develops when both alleles are lost. That means this is something different. Okay, I will not go into the detail of this. Okay, counseling. Okay, what counseling you will give to your patient? No treatment. Okay, as for most of the genetic disease, there is no treatment, but prenatal diagnosis is advisable when a parent is suffering from neurofibromatosis, and when the wife get pregnant, or uh, she is pregnant, then one should go for the investigation whether the baby will also suffer from neurofibromatosis or not. Okay, let us see this person who is suffering from neurofibromatosis. You can see the small pea-like tumors on the skin. Okay, so this is the neurofibromatosis. Okay, I think this completes the today's lecture. Thank you for watching.